Next speaker is Erin Kirk. Erin grew up in Lexington, Virginia, and she spent her childhood playing outside and going out in the field with her parents, biologists, testify, to help them do field surveys and fish surveys. Her dream job would be working with wildlife rehabilitation centers. She loves helping animals, even if it's picking up and bringing home strays. This summer, she is marrying into the military, Emory and Henry alum, chemistry, but we're okay with that, and moving out to California. This will allow her to travel and to see the world, and her goal is to go on to graduate school and then be able to find a job working with wildlife and field biology. Erin.
Thanks. Did you pull it up?
Hi, my name is Erin Kirk, and today I will briefly be talking about the research I do here at Emory and Henry College, which is the phylogeography of Montane salamanders in Southwest Virginia. And phylogeography, all that means is it's a study of historical processes that could be responsible for the geographic distribution of individuals today. So my entire life, I lived and grew up around the Appalachian Mountains. And as even as like a young child, I would be out in the rivers and creeks, and I would collect salamanders and snakes and other little critters like that. So I knew that I really wanted to do research that involved being outdoors. And I was like, what better to do my research than in the Appalachian Mountains, which is basically out our back doors. So then I was like, okay, I know I want to do that. So doing some research, I found out that Whitetop Mountain and Mount Rogers both exhibit mountaintop islands, which means the top of the mountaintop, the habitat is significantly different than the habitats on the base of the mountain. I was like, that's really neat. So I started doing a little bit more research, and I started looking into it. And I found out that these mountains contain relics of the previous glacial period, the Wisconsin Glaciation, which occurred about roughly 22,000 years ago. And so I started looking into that, and I was like, that is really cool. Like, I didn't know that whatsoever. And I started digging up some more research and found out that when the Wisconsin glacial period was here, that all of these creatures from up north, they either went extinct or they pushed down to more southern areas because it was too cold for them. And then when the glacial period receded back up north, so did either these animals or like the salamanders who needed these cooler temperatures got stuck up on these mountaintops because that is where their habitat was. So these salamanders are pretty much restrained to those high elevation peaks. So I was like, okay, great. I have my research. Let's go collect DNA now. Started going out there realizing that the most common way to collect DNA from salamanders is a super invasive tail clipping, which means you're just like pulling off the tail of these salamanders. And I was like, that doesn't make any sense because when you're looking at endangered or threatened species, you want to use a non-invasive method. I'm like, why would we want to harm all these salamanders when they're already threatened? So I started looking into it and found out that, hey, we can use this non-invasive epithelial body swabbing. And that's just taking their skin cells by swabbing so it doesn't hurt them, and it's really non-invasive, and it's much better to use than the tail clippings. And so that is what I'm going to be focusing my talk on today, is this non-invasive epithelial body swabbing of these salamanders versus the invasive tail or toe clippings. So my target species are these four. And the one on your guys' left is a Plethodon montanus, then it goes Plethodon cinereus, the Plethodon welleri, and the Dysmnatheus wrighti. And these four species are located on both the Whitetop Mountain and the Mount Rogers. And then I also wanted to look at the Elk Garden area in between these two as a corridor for these salamanders. So these are the four that I look at. So a bit about methods. So obviously I have to go out and I do a lot of field work, which I love. I like it more than lab work. So I enjoy doing this stuff. And so I go out in the field. I will sometimes even camp out overnight. That's the fun part. I'll collect these salamanders. I don't take any voucher specimens because that would defeat the purpose of preserving these endangered species. So I would swab them, I would return them to where I found, either under a rock or a log, and then I would take that DNA that's on a Q-tip, and I would come back to the Emory and Henry labs, and then I would perform a DNA extraction using a DNeasy blood and tissue kit. And so that's just extracting the DNA from these Q-tips and putting it into little tubes. And from there, I quantify this by using a nanodrop. And by doing that, that is just telling me the concentrations of DNA that I have in these tubes. So the higher the number in these tubes, the more DNA I have. And the more DNA I have, the better it is. So then after that, I do a PCR amplification. And that's just amplifying all this DNA that I have in genes. And so I then, from there, with it amplified, I can then go on and visualize it on a 1% TBE gel. And that is just looking at, that's taking those numbers and putting it into picture form, so then I can really look at what those numbers mean. So I know this is a lot on this slide, but I kind of need it all so I can show you guys what I'm talking about. So if we look at this top chart, that is all tail clippings. This middle chart is the epithelial swabs. And all of this is done on museum specimens. And I did that because I wanted to show you guys that if we can use, if this technique works on those already stored museum specimens, that we wouldn't have to continue taking voucher specimens or killing more live salamanders that would harm the population level. So this is all done on museum specimens, because if the swabs work on this museum specimens, then that's a really good starting spot. So if we can look, this in the dark blue, that are museum specimens that were previously stored in ethanol. The ones on the right were previously stored in formalin. As we can see, the formalins did not work. So the numbers we're looking at are these first row, the concentration. So there, and then those ones. Ignore all the other numbers. Pretty much we just want you to look at the concentration. So the formalin did not work. So from here on, I just used ethanol museum specimens. If we compare the tail clippings, that number compared to that number, we see the tail clippings work significantly greater. So that kind of bummed me out, because I was like, okay, well, that kind of defeats my research there about using these swabs on these salamanders. So I was like, all right, there has to be another way. So what I did was I took those 4 specimens there, and I did a vacuum centrifuge. And all this is taking those tubes, and then I spin them down really fast, and then I rehydrate them to a concentration that I want. So this just multiplies, giving me a higher yield of DNA concentration versus the liquid that's in there. And as you can see, these numbers right there, which are swabs, are equivalent or roughly equal to the tail clippings. So that really worked well. Then I ran these samples on a 1% TBE gel, and here's that gel there. As we can see, the top are swab and the bottom are tail clippings. They both showed up very nicely, and they both worked. So then I took 2 samples that I had, and I sent them off to get sequenced, which this is what I got back when it was sequenced. The top is tail and the bottom is swab. And you want to send them off to get sequenced because I need a reliable source to verify that the salamander I actually caught is indeed a Plethodon welleri, or it is a Cinereus. Because since I'm not taking those voucher specimens, I can't verify that that is what that specimen is. And I need some way to tell that that salamander is a weller or something. So as you can see, the tail is slightly cleaner. There's none of these little fuzzy pieces down there. However, the swab does show up just as well. And if you line them up, you can tell because of these peaks, they line up. They are from the same individual. So this swab and tail were from the same salamander that I took. So that actually really pushes my research forward and shows me that, hey, swabbing does work. This epithelial body swabbing works just as well as the tail clippings. So my next step in this research is to actually run a gradient on the PCR, and I really need to do that first because if my gels aren't clean enough, as you could see in the previous gels, there's this non-specific banding. And if it has that non-specific banding, I cannot send it off to get sequenced. It just won't. It's unable to be sequenced. And without it being sequenced, I can't verify that that salamander is what it is. So I really need to run a gradient in order to clean up that sequencing, and then I can send all those specimens off. Then when I send them off, I can actually verify what they are, and then run into and do my overall project, which is the phylogeography of it, because then I can take those sequencing samples and look at them and then compare them and then send them off to another computer database, and it will tell me the actual historical background of these salamanders, which is really cool, and that's what I actually want to do with this research. And then another huge aspect of just doing this epithelial body swabbing is that it allows you to collect enough DNA from this Amphibian Chytrid fungus. And I don't know if anybody has heard of the Chytrid fungus, or there's two main types right now, but it's really detrimental to pretty much all, not only salamanders, but like frogs and toads as well. And it can wipe out entire populations of these amphibians. So the epithelial body swabbing allows us to get enough DNA from the individual, and then we could run tests and to see where this Chytrid fungus is located. And if we know that, maybe we could potentially find out how we can prevent this Chytrid fungus from spreading, how we can stop it, just because we don't want the Chytrid fungus to continue spreading. Whereas the tail clippings, you don't get enough DNA from the Chytrid fungus to determine if it's Chytrid fungus. So another huge environmental aspect of this epithelial body swabbing is that you can then further look at the Chytrid fungus. I would like to thank Dr. Argyros and Dr. Brezoir for their help with this project and coming up and helping me with all my problems that I have run into. I would also like to thank all the grants I've gotten. Without their support or help, this project would not have been able to take place. And what's up?